ZeroPM we have an amazing group of students who are working on lots of different topics. You can see they're hanging their posters up now. So let's go and listen to what they have to say about their work.
Hi, I'm Ellise Suffil. I'm a postdoc at the University of Vienna working on ZeroPM. So the role of social sciences in any project involving risk, be it from chemicals or somewhere else, is to understand how people reason about that risk. So they're doing something called risk perception, which is different to risk analysis. If you imagine you're an expert in chemistry, you can say what's the harm of this chemical and what's the likelihood that someone's going to be exposed to that chemical, and this is called risk analysis. But if you're a normal person, you don't have that information available to you, so you do something called risk perception. Now, risk perception is quite different because it has a lot of psychological factors associated with it. So one such factor is vulnerability. Who is going to be affected by this risk? If it's children, if it's the elderly, then people will assume that risk is higher than it actually is. Another example is, for example, the fear of the unknown or dread. So if you imagine flying, people are quite scared of getting in a plane, but actually the risk associated with getting in a plane is a lot lower than getting in a car. But most people will still get in their car, drive to work and back every day. The fear with the plane is that we feel like we're out of control, right? We can't directly fly the plane ourselves if something goes wrong. So when normal people are perceiving risks, there's lots of other factors at play. And our job is to measure these risk perceptions, to understand why people feel that way, and in key cases to try and understand and correct misconceptions. So for example, where people may be worried about things that aren't as dangerous as they feel they are, or to actually inform them that the risk is there that they didn't know was there. For example, in the case of PFAS, basically we need to inform people what PFAS is, what they can do about it, and what sort of policies they can support to help in reducing PFAS pollution going into the environment.
Hi, my name is Romain Figuire. I'm a PhD student at Stockholm University in the Department of Environmental Science, and I'm working on the implementation of the essential use concept in the REACH regulation. What I did is that I looked at previous decision in the authorization and restriction processes under REACH and looked at if it relates to the essential use components. And what we found out is that it relates mainly on the availability of alternative and not so much on the necessity for health and safety. And what we also found out is that there is no need to change completely the authorization and restriction processes to implement the essential use concepts in REACH.
My name is Emma Palm. I'm a PhD student at the University of Luxembourg, and my poster today is about Shiny TPs, which is an application that I've developed for curation of transformation reactions from PubChem text sections. And the idea of this is that we should be able to get more information about what transformation reactions can occur in metabolism or water treatment, so that this can be used in further workflows for, say, suspect screening or database development and so on.
Hi, my name is Sivani Baskaran. I'm a postdoc in ZeroPM with Work Package 5. I'm based at the Norwegian Geotechnical Institute in Oslo, Norway. The work I'm presenting here today is how we can use Bayesian approaches to identify a consensus value for the KOC, or the organic carbon-water partition ratio. And this ratio is used to identify chemicals that are considered mobile in the environment. So chemicals with a log KOC value less than 2 are considered very mobile, and chemicals with a log KOC value less than 3 are considered mobile. And this Bayesian approach allows us to combine a variety of different chemicals, chemical data from different sources, and including experimental, including estimated values, to determine a single value that we can use for these threshold assessments.
Hello everybody, my name is Abishek Laxmanan, and I work at Fraunhofer ITEM with Dr. Sylvia Escher and Jenny Irwan. So we belong to the work package 6, and our role at the ITEM is to work with the toxicokinetics or the kinetics data. So our aim is to use the in silico and the in vivo data set, and then our process on the whole is termed as eViva, which means in vitro to in vivo extrapolation. And then we go to the lab, or we have our colleagues who work in the lab, and then we get the in vitro parameters, which has to be then corrected based on an in silico based model, which is termed as vivid model, and then we make the correction, the dose response versus the concentration, and then we have our PBPK model or PBK model developed from Fraunhofer ITEM, and this data serves as the input to the PBK model, and then later on we get the output, and the PBK model on whole has lots of mathematical equations and connected to the different parts of organs of our body. Now I handle over to Jenny Irwan over the compound selection and also the read across approach.
Yeah, so hello, my name is Jenny Irwan. I'm also a colleague from Abishek from the same institute. And here I work for the compound selection. So we have triazine, triazole, and PFAS, the three classes of PM compounds or persistent and mobile compounds. We selected these compounds because they are known for this PM character. And then here, for example, it's the first class, it's the triazine class. For the triazine, we have 4 selected compounds. It's the cyromazine, amylide, melamine, and atrazine. And here are the data-rich compounds which will be measured in the lab. And these are the compounds that are data poor or don't have so many data. And with the help of this data from data-rich compound, we can predict the toxicity of the data-poor compounds. And here we also not incorporating the not only the parent compounds, but we also have here the metabolic pathway of cyromazine. The cyromazine will be then degraded to melamine, and both cyromazine and melamine are part of the selected compound. It means we can incorporate not only the parent compounds, but also the metabolites or the degradation product from the selected compounds. And then not only that, we also have this toxicological profile of the PM compounds from the triazine, triazole, and PFAS. And these 3 compounds have same target organs. It's the liver. So liver is the main target organ of these PM compounds, and not also liver. There's also another effects from other organs like kidney, spleen, and testes, and heart, and so on. And these information of the toxicological profile will also be considered and also will be incorporated to our prediction of the data poor compound. That's all from our side, from Fraunhofer ITEM. Thank you.
Hi, I'm Lukas Leismeister. I work at TZW Water Technology Center in Karlsruhe in Germany. My research as a doctoral student deals with PFAS removal from drinking water by ion exchange processes, and specifically in ZeroPM. I'm investigating a hybrid process consisting of granular activated carbon filtration and anion exchange to remove perfluoroalkyl acids, which are terminal degradation products of many PFAS. The idea of the combination was developed because perfluoroalkyl acids with short alkyl chain lengths do not absorb all too well to activated carbon and have quite early breakthroughs in the filter compared to perfluoroalkyl acids with long chain lengths. And the new EU Drinking Water Directive sets quite strict threshold values that are much more strict for the short chain perfluoroalkyl acids than before, reducing the operating time of activated carbon filters by a lot. And now by placing a anion exchange process behind the activated carbon filter, dealing with the short chain compounds with early breakthroughs, can extend the operating time of the activated carbon filter again. The resins I want to use is that they are regenerable, so they can be reused and be in operation for quite a long time. And one of the challenges here is to find appropriate resins that have decent absorption capacity towards PFAS and are regenerable at the same time. And on the poster, I'm presenting some very interesting results from experiments with a strong base anion exchange resin that has shown decent absorption and was regenerable with sodium nitrate as a regenerative agent in batch experiments with continuous or consecutive adsorption and regeneration cycles, and also in a laboratory column experiment.
ZeroPM, Zero Pollution of Persistent and Mobile Substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under Grant Agreement number 101036756.